Hello, I'm Emilie Plantamura. I'm a Deputy Chief Medical Officer at MaaT Pharma. Today I'm going to talk about MaaT 33, a drug that is tested in the PHOEBUS phase 2 trial. So briefly, MaaT 33 is a pooled microbiome ecosystem therapy. So it consists of fecal material coming from several donors that are put together in order to increase the diversity of the product. The product is lyophilized and then is put in capsules. So this is an oral form that can be taken orally by patients. This allows to have, it's the easiest way of administration for patients. So the PHOEBUS trial is a phase 2b European randomized controlled trial versus placebo. So the rationale of this study started when we read the paper from Jonathan Peled and colleagues from the Memorial Sloan Kettering Center. They showed that indeed it was a retrospective study with a high number of patients, and they showed that the higher the gut microbiota diversity is in Allo-HSCT patients at the time of the neutrophil recovery, the higher the survival is. And this was very interesting, and the magnitude of the effect was really high when they compared low diversity versus high diversity patients. And so the hypothesis behind this is that when the patient has a high diversity of the gut microbiota, it may prevent acute GVHD following Allo-HSCT. It may also prevent infections because a gut microbiota is kind of physical barrier in the gut, and the gut is one of the main origin of infections. So it's also hypothesized that the gut microbiota may have an impact on immune reconstitution and maybe on relapse, but this is really a hypothesis, and that's what we want to test in the PHOEBUS trial. So the PHOEBUS trial is a randomized controlled trial versus placebo. So the study plans to include patients that are about to receive Allo-HSCT. So the criteria are adult patients that will receive adult patients over 50 years old that will receive reduced intensity or reduced toxicity conditioning regimen in order to have an homogeneous population. And we exclude cord blood transplantation, but otherwise the criteria are very broad. We have some exclusion criteria regarding eligibility to Allo-HSCT and also some specific criteria related to fecal microbiome therapy, such as gastrointestinal diseases. But otherwise, this is a trial. MaaT 33 is aimed to be considered as an add-on therapy in addition to the standard of care, which means we stick to the standard of care in terms of for Allo-HSCT patients. So we do not change the practice of physicians. The study is ongoing in Europe, in France, Germany and Spain, and is about to be open in other countries. We had a first data safety and monitoring board very recently evaluating the safety of the trial, and of course, as of today, there is no safety signal and the trial could continue without modification. So the design of the study, there are two treatment phases in the trial. First, before the Allo-HSCT, the aim is to restore the gut microbiota of these patients, because most of them will have received previous cycles of chemotherapy, previous antibiotics, and it has been shown that the gut microbiota diversity of this patient before Allo-HSCT is already impaired. So the idea here is to restore the gut microbiome before Allo-HSCT to ensure that at the time of the neutrophil recovery, they will have a gut microbiota as high as possible. And then following neutrophil recovery, because we don't treat during aplasia of this patient for safety reasons, After the aplasia recovery, we start the second treatment phase, so around day 18, day 25, it depends on the patients, and the treatment will continue up to month 3. This is a critical period for allo-HSCT patients because this is the period where acute GvHD occurs, where most of the patients have infections, and so that's why we try to and also they receive antibiotics during this period. And by taking MaaT 33, by restoring their gut microbiota, we aim to counteract the effect of antibiotics, and we aim also to prevent all those complications that are found after allo-HSCT. So the aim of the study is to prove that with the gut microbiota restoration with MaaT 33, we are able to restore the survival based on the rationale from Jonathan Peled and colleagues. So the primary endpoint of this study is the evaluation of the overall survival at 1 year, and we aim to demonstrate 10% increase of survival in those patients.